Sometimes for a cell to function, we want particular proteins in the cell to be in a particular area, or we want the movement of those proteins to be restricted and not to be floating all around within the phospholipid bilayer. So this experiment is showing that some proteins can diffuse within the cell membrane. So in this experiment, you would have a mouse cell where they've labeled a protein with rhodamine, which is red. And this is a transmembrane protein, so a protein that passes across the cell membrane. And now each protein glows red under fluorescent light. We need the fluorescence because you cannot otherwise see the proteins. We have another cell, in this case it's a human cell, where there is a membrane protein that is labeled with fluorescein, which is green. You can take these two cells and pass a current across them and fuse them. When this happens, you get a hybrid cell that is part mouse cell with the red proteins and part human cell with the green proteins. If you took this cell and looked under a fluorescence microscope, you could only look at one color at a time. You would look at red and you'd see half the cell glowed red and that the corresponding half or the other half would be green when you switch to a green filter set. This is at 0 minutes, so right after you fuse the two cells. If you wait 40 minutes though, for these particular membrane proteins, you would see a mix that they diffuse across the cell membrane. So proteins that came in from the original human cell that were tagged green are now throughout the membrane. Same thing with the rhodamine red labeled membrane proteins are now scattered through the cell membrane. However, not all proteins can diffuse across the cell membrane, and there are a number of different things that can restrict them. So we are looking here at 4 of those categories of anchors or ways to sequester protein. The first is that proteins within the cell membrane can bind to the cell cortex, right, that underlying cytoskeleton structure. If they're stuck to the cytoskeleton, that cytoskeleton that's right next to the cell membrane, they won't move. Similarly, proteins can be anchored to the extracellular matrix, right? So again, this is extracellular matrix has protein fibers in it. They're very strong. If the proteins within the membrane are stuck to the extracellular matrix, they're not going to move around because the extracellular matrix is stuck in place. Also, cells, if they're adhered to one another via proteins, there won't be diffusion. So this particular protein here at the top is stuck to the protein in another cell. And because of that attachment, it can't diffuse. Another way of restricting movement is something called diffusion barriers, which would be here, these little boxes. An example would be tight junctions, which are very tight seals between two cell membranes. In this case, it's a barrier. It doesn't mean that the proteins cannot move. It just means they can't move past the barrier. So the green proteins can move along on this side, which would be the apical side of the cell. And then the blue proteins can diffuse within the region below the diffusion barrier. We'll see this type of diffusion barrier is at work in epithelial cells of the gut. So here's a little closer look at that. We can see the apical domain. If this is an epithelial cell of the intestine, this would be facing where I have the laser pointer, the interior of the intestine where your food is. We would come through across the basal lamina, and there would be a capillary here to collect the nutrients. And here's that tight junction that serves as a diffusion barrier. We're going to find in chapter 12 that proteins in the apical surface that transport glucose work in a different way than proteins in the basal membrane that transport glucose. So we will see this tight junction and this diffusion barrier play a role in the functioning of our digestive system in the next chapter. We can measure how quickly proteins are moving around within one side of the bilayer, this would be called lateral mobility, through a process called FRAP.
The lateral mobility of membrane proteins can be measured in living cells by FRAP, which stands for fluorescence recovery after photobleaching. For this purpose, membrane proteins are often expressed as fusion proteins with the green fluorescent protein GFP, and observed with a fluorescence microscope. A selected area of the cell is then bleached with a strong computer-controlled beam of laser light. Those membrane proteins that are not anchored and therefore can diffuse in the plane of the membrane quickly exchange places with their neighbors and fill back in the bleached area. From the rate of this fluorescence recovery, the diffusion constant of the protein can be calculated. Here, GFP is fused to a membrane protein that lies in the membrane network of the endoplasmic reticulum. After bleaching, we observe quick recovery of the fluorescence, showing that the protein is very mobile in the plane of the membrane. The same experiment can be repeated using a protein that is firmly anchored and not free to diffuse. Here, we observe GFP fused to a protein of the internuclear membrane that binds tightly to the meshwork of the nuclear lamina. After photobleaching, no fluorescence recovery can be seen over the same time frame.
Another aspect of membrane proteins is, like glycolipids, they can have sugars attached. And there are two categories of membrane proteins with attached sugars. The first being glycoproteins. Here is a transmembrane glycoprotein with short oligosaccharides attached. And here is a transmembrane proteoglycan. It's also a protein with attached carbohydrate chains, but these are they have one or more long polysaccharide chains. Okay. So together, the glycoproteins, the proteoglycans, and the glycolipids create a carbohydrate-rich layer right outside the cell. This carbohydrate-rich layer has many functions. Here are a few examples of why a cell would have a carbohydrate layer. Because you have a lot of sugars kind of on the outside of the cell, this creates an osmotic gradient, which can cause kind of a lubrication that helps protect the cell from mechanical and chemical damage. It can also be a way that cells distinguish one another. So they recognize cells they might want to adhere to, for example. In a moment, I'm going to show you a movie of carbohydrates in a white blood cell that are used in a binding interaction with the cells, the endothelial cells of the blood vessel, and is helpful in those white blood cells exiting the blood vessel and going to the site of infection. Another example of where a carbohydrate layer plays an important role in cell function is during fertilization. For example, in sea urchin embryos, they have carbohydrates, kind of an egg jelly is what it's called, outside of the egg. For sperm to bind, it has to recognize the specific carbohydrates surrounding that egg. And different species of urchins have different carbohydrate layers. So the specific carbohydrates outside of the egg vary by species of sea urchins, so that you have a very specific fertilization of a sperm from the correct sea urchin species with an egg from the correct species, right? Because they have external fertilization in the ocean, in the water. You need a mechanism or a way to have proper fertilization, and carbohydrates are important in that process. This movie shows this process both in an animation and also in a live animal.
Leukocytes are white blood cells that help fight infection. At sites of injury, infection, or inflammation, cytokines are released and stimulate endothelial cells that line adjacent blood vessels. The endothelial cells then express surface proteins called selectins. Selectins bind to carbohydrates displayed on the membrane of the leukocytes, causing them to stick to the walls of the blood vessels. This binding interaction is of sufficiently low affinity that the leukocytes can literally roll along the vessel walls in search for points to exit the vessel. There, they adhere tightly and squeeze between endothelial cells without disrupting the vessel walls, then crawl out of the blood vessel into the adjacent connective tissue. Here, leukocyte rolling is observed directly in an anesthetized mouse. The up and down movement of the frame is due to the mouse's breathing. Two blood vessels are shown. The upper one is an artery with blood flowing from right to left. The lower one is a vein with blood flowing from left to right. Leukocytes only adhere to the surface of veins. They do not crawl out of arteries. Some leukocytes are firmly attached and are in the process of crawling through the vessel walls, whereas others have already left the vessel and are seen in the surrounding connective tissue. When the blood flow is stopped temporarily by gently clamping the vessels, we can appreciate how densely both vessels are filled with red blood cells. Red blood cells do not interact with the vessel walls and move so fast under normal flow that we cannot see them. When the blood flow is restored, some of the leukocytes continue rolling, whereas all non-interacting cells are immediately washed away by the shear.
So here's a summary slide of what you just saw in that video, where we have adhesion that's going to occur between the neutrophil and the lectins and the endothelial cells lining a blood vessel. And in the video, you see them rolling along until they find a space between the endothelial cells where they can leave the blood vessel and then go to the site of the infection to help clear it out. So that's going to be the end of chapter 11. From here, we will move on to chapter 12.